Okay, so we start with a discussion on the MCQs that were given to you as a quiz. So the question says that an experiment was carried out to compare the effect of pH on the activity of an enzyme that was in solution and the same enzyme that had been immobilized on a gel. All other variables were kept the same. So you have two graphs. The graph which is shown with a circle is the immobilized enzyme wala graph, and the graph which is shown with those small dot squares is the enzyme in solution. Okay. Now the question is, which statement explains these results when the enzyme is immobilized? The primary structure has changed. So guys, do you think that any immobilization of the enzyme can change the primary structure of a protein? Yes or no? Yes, Alia. No, primary structure is the sequence of the amino acids in an arrangement, right? Secondary structure has changed. Do you think that when we immobilize an enzyme, अच्छा एक तो ये कि results भी हमें नजर आ रहे हैं, तो what we can see is that we can see that the enzyme which is immobilized is more active over a wider range of pH, and the enzyme which is non-immobilized or the normal wala enzyme is active within a narrow pH range. हमें graph को देख के apparently ये नजर आ रहा है. So of course from this graph ये तो कहीं से भी नहीं बनेगा आंसर कि the primary structure has changed. Do you think the secondary structure has changed? Yes, Alia, Amna? No, the secondary structure has not changed. ठीक है? And then the tertiary structure has changed. Do you think that?
So I'm not sure about this one because it could be the quaternary structure too.
Huh. Basically, the tertiary structure has changed, right? Because the changes in the pH or the changes in the temperature, it affects principally the tertiary structure. That structure which is responsible for the extensive coiling and the folding of a globular protein like an enzyme. All globular proteins, whether they are hormones, they are enzymes, or they are antibodies, or it is hemoglobin, we call them globular because of their extensive folding and coiling. और जब हम उनकी बात करते हैं कि the effect of temperature and pH on them, that's principally hitting the tertiary structure. So of course, जो भी हमें change नजर आ रहा है, discrepancy नजर आ रही है between how an immobilized enzyme is behaving over a certain pH range and how a mobilized enzyme is behaving, दोनों cases में कहीं ना कहीं आके उनकी activity रुक जाती है. तो क्यों रुकती है? किस चीज की वजह से रुकती है? Because the tertiary structure has changed, not the quaternary structure. Quaternary एक अलग चीज है. Quaternary का मतलब है कि a protein made up of more than one polypeptide chain. इस वक्त उसकी बात नहीं आ रही. और usually जो enzymes होते हैं, usually they have a tertiary structure. Quaternary भी हो सकता है, लेकिन ज्यादातर they have a tertiary structure. And when we talk about the effect of pH and the effect of temperature on enzyme, तो हम tertiary structure की बात कर रहे होते हैं. Is this clear and understood to both of you?
Yes.
ठीक है? अच्छा. I think, should we revise the thing? एक-एक करके, should we go through it? Okay. Alia, beta, you tell me. What is the answer of MCQ number 2?
Option B.
B, very good. Low, It's okay, Alia. Your connection is very much broken, but I know that you know the concept that if anything has a low Michaelis-Menten constant, it means that the affinity of that enzyme for its particular substrate is high. And if anything has a high Michaelis-Menten constant, जिसको हम high Km कहते हैं, Km भी कह सकते हैं, Michaelis-Menten constant भी कह सकते हैं, then this means that the affinity of that enzyme for a particular substrate is low, right? Acha. The diagram shows a metabolic pathway. Amna, you tell me what will be the answer to this question?
B.
B, correct. Substance X will increase in concentration. ठीक है? जो non-competitive inhibitor होता है ना, अगर हम वो देंगे, तो in that case, substance X will increase in concentration. ठीक है? अच्छा, and then moving on to this much, this one. Which statement is true of all enzymes? Yes, Amna. 4.
This is D.
D, yes, sub enzyme ke liye, because all enzymes are not inactivated at low pHs. All enzymes don't catalyze the breakdown. They might catalyze the making of some molecules or the detoxification of some molecules. And all enzymes are not denatured at 60 because there are some enzymes which are found in the bacteria that live in hot springs. Hot springs का temperature 80 से लेकर 100 तो होता है. तो वहाँ पे जो bacteria रहते हैं, they also have enzymes, and of course their optimum temperature is much more than 60 degrees Celsius. Okay, now this one. Let me hold it like this. Okay, so Alia, can you tell me what will be the answer to this? Question number 5. Which graph shows the change in the concentration of the enzyme substrate complex? Yes, Alia.
Miss, I think it's option B.
Option B.
Miss, option B.
No, not option B. What did you choose, Amna?
It's D.
D.
Yes, miss.
5D. Okay, let's look at this question. Let's study. The answer is incorrect actually for both. It's neither D nor it's B. Let's see what it is. The quantity of an enzyme was added to a quantity of a substrate. The graph shows the changes in concentration of the enzyme, the substrate, and the enzyme substrate complex and the product over time. ठीक है? अच्छा. Which graph shows the change in the concentration of the enzyme substrate complex? यानी जैसे जैसे time गुजरता गया, तो जो change हुआ concentration of enzyme substrate complex, वो कौन सा वाला ग्राफ शो कर रहा है? आपको कोई ऐसा ग्राफ चूज करना है जिसमें कि in the beginning the concentration of the enzyme substrate complex was low. As time is passing, it's gradually increasing. ठीक है? और उसके बाद क्या होना चाहिए एंजाइम सबस्ट्रेट कॉम्प्लेक्स को? आहिस्ता आहिस्ता फिर एंजाइम सबस्ट्रेट कॉम्प्लेक्स को कम होना चाहिए. यू नो व्हाई? बिकॉज इट विल नाउ बिकम एन एंजाइम प्रोडक्ट कॉम्प्लेक्स. पहले वो सबस्ट्रेट से बाइंड करता है, फिर सबस्ट्रेट को प्रोडक्ट में कन्वर्ट करता है, और फिर वो पहले सबस्ट्रेट से बाइंड करता है, देन देयर इज एन एंजाइम प्रोडक्ट कॉम्प्लेक्स, और फिर प्रोडक्ट्स को वो लीव कर देता है. तो व्हाट डू यू थिंक? Which graph shows you कि शुरू में एंजाइम सबस्ट्रेट कॉम्प्लेक्स कम है, आहिस्ता आहिस्ता बढ़ के पीक पे जा रहा है, और थोड़ी देर बाद जो है वो वापस इट्स कमिंग डाउन टू इट्स ओरिजिनल concentration. So what graph is that?
A.
A. Acha, for this, let's look at, it's good that we should revise this because there is a slide over here on enzymes जो कि आपको सही से इसके बारे में बताती है, and that is this slide. Yes, this one. Have a look. First, you have a substrate entering the active site. Then you have an enzyme substrate complex. ठीक है, इसका मतलब यहाँ पे the enzyme substrate complex बिल्कुल जीरो था. आहिस्ते आहिस्ते टाइम के साथ बहुत ज्यादा हो गया. ठीक है, जब ज्यादा हो गया फिर क्या बन गया? अब ये खत्म हो जाएगा. अब क्या बन जाएगा? एंजाइम प्रोडक्ट कॉम्प्लेक्स. फिर एंजाइम प्रोडक्ट कॉम्प्लेक्स भी कम हो जाएगा, और फिर क्या बन जाएगा? प्रोडक्ट्स अलग, एंजाइम अलग. नाउ डू यू गाइस मेक अ कनेक्शन बिटवीन दीज टू?
Yes, miss.
ठीक है, तो this it was, you know, we always learn through our mistakes, so it was good that you pointed out this mistake. ठीक है, let's see this. Okay, so I'm not reading out the question. Diagrams X, Y, and Z represent these enzyme molecules and their active sites. State the letter X, Y, or Z that most accurately represent the enzyme DHPS. Okay, so which letter represents that? Yes, Alia.
Miss, letter X.
बिल्कुल सही, letter X सही. अब हम आते हैं इस क्वेश्चन पे, and that's the last question. कि क्या है? Using the information in figure 6.1. Figure 6.1 को देख के बताना है. ठीक है, using the information in figure 6.1. बहुत सारे बच्चों ने टेस्ट में भी इसलिए मिस्टेक की थी कि उन्होंने यहाँ से पिक्चर से इंफॉर्मेशन नहीं ली और उसके बगैर बताया है. So using the information in figure 6.1, you are supposed to explain why sulfonamide acts as a competitive inhibitor of DHPS. Use this information and tell me why sulfonamide acts as a competitive inhibitor for DHPS. And there are 4 marks. So then you need to give me 4 reasons from the picture. Alia, आप मुझे 2 reasons बताओ, and Amna, you can tell me 2 other reasons. Yes, Alia.
Miss sulfonamide, it has a complementary shape to the active site of the enzyme, so it can bind to it and
Correct.
prevent PABA from being catalyzed.
Correct. ठीक है. Complementary shape, so it can bind to the active site. ठीक है, तो मतलब आपने 2 जो इसके आंसर्स हैं ना वो बिल्कुल सही बताएं. 2 points. Now, Amna, your turn. Now, since she has chosen the very easy points, so you are now left with the difficult points, and your points you will choose from the diagram. Ji, aap batao.
Since, like she said, they have a similar structure, but they're not exactly the same. So the enzyme cannot act on the sulfonamide. So the PABA is left to wait until the sulfonamide just leaves the structure, active site.
No. See, what mistake are you doing? Once again, let's see the question. Explain why sulfonamide acts as a competitive inhibitor. ये बातें तो उन्होंने बता दी कि ठीक है ये इसलिए competitive inhibitor की तरह का है because it has a similar shape to the substrate. It can't bind to the active site. ठीक है ना? तो इसलिए block कर दिया उसको active site को. Then other similarities which you can see from the picture is you will say both these have a benzene ring. Don't they have a benzene ring? Both of them have an amino group, NH2, sulfonamide and this. Yes, Amna, I can't hear you.
Yes, miss.
Did you understand what I'm saying?
Yes, they have the same groups.
They have the same groups. They have, both of them have amino groups. Both of them have a 6 carbon ring. या आप कह सकते हो कि जो है वो both of them have a benzene ring. ठीक है? Similar है ना? बहुत ज़्यादा similar है. इसी वजह से जो है it is acting as a competitive inhibitor. ठीक है? नहीं समझ आ रही? ये समझ आ गई?
Miss, isn't that the first point?
First point. Acha, wait. Let me show you the mark scheme. Have to stop, share it, and then show you the mark scheme. Okay. Now have a look. Okay. Wait. Now, Alia had pointed out these points. Okay? The substrate, which is PABA, and the inhibitor have same shape. Don't they have a same shape? आपने अगर आप same shape कह दोगे ये एक mark हो गया, right? And then you will say, so hence they are able to bind and fit into and block the active site because the shape is complementary to the active site. ठीक है, ये 2-3 points तो हो गए. But then if this question is carrying 4 marks, तो आप क्या लिखोगे कि you have to pick up similarities from the diagram ना. तो आप diagram में देखोगे कि let me see कि what is the diagram telling me. The diagram is showing me that both of them किस तरह से similarity है कि both of them have amine groups, both of them have a 6 carbon, or you can even say they have a benzene ring. ठीक है, बस यही बात अब समझ आ रही है. Is this clear?
Yes, miss.
Right? Acha. Let's go back to the, acha, this is finished. Now I have another difficult quiz, and that quiz is on cell structure and enzymes. Let me see how many, just a minute. Okay, so last time we had done fluid mosaic model, and I'm assuming that both of you do not have a problem in understanding this, right? Just a very fast recap that the cell membrane is a living structure, 7 nanometers wide, fluid and mosaic. Fluid because of the phospholipids which are diffusing in their own monolayers. Sometimes some proteins are also diffusing. Some of them are anchored, some of them are diffusing. Mosaic means having a dotted or a tile-like appearance because of these four structures. Namely, the phospholipids themselves are giving it a mosaic appearance, the proteins, the cholesterol, the glycolipids, and the glycoproteins. And if you guys remember, we had discussed this in detail. Ye bhi humne kiya tha, and we did this MCQ also. So I hope there is no problem as far as the understanding of this concept is concerned. अगर कोई है तो आप मुझसे पूछ लो, वरना आई एम मूविंग फॉरवर्ड जी. नो प्रॉब्लम. आमना?
It's clear, miss.
Clear. अच्छा. Now, when we look at the cell membrane, the examiner is going to confuse you and ask you different questions. The first thing that he will ask you he will say, what is the core or what is the majority of the cell membrane made up of? So your answer should be that the core of the cell membrane is made up of fatty acids, the major part. So to cut short, we will say that primarily the cell membrane is hydrophobic. Primarily, it is repelling the water-soluble polar substances and sort of welcoming the non-water-soluble, non-polar fat-soluble substances. ठीक है? There are only two substances जो कि cell membrane कभी भी stop नहीं करती उन substances की movement. Those are water and carbon dioxide. अच्छा, water जो है वो तो है ही polar, उसको तो रोकना चाहिए cell membrane को, but it doesn't stop it. And if the examiner asks you the reason, you will say that it's because water is a tiny molecule. It's very small, so it can easily push these fatty acid tails and it can make space for itself. ठीक है? Second molecule जो है वो आपका carbon dioxide है, वो तो non-polar ही है. It's a small non-polar molecule, so it can also easily pass through the cell membrane. Besides these, no polar water-soluble molecule can pass through the cell membrane at all unless and until it has a help through these proteins, which you can see on the diagram. ठीक है? अब एक-एक करके मैं आपको इन सबके effects बताऊंगी, right? The role. अच्छा. Phospholipid bilayer, phosphate heads being water soluble, facing the exterior of a cell and the cytoplasm face of the cell. Core of the cell membrane made up of the hydrophobic fatty acid chains. Scattered amongst these fatty acid chains, we have cholesterol molecules, which also look very similar to fatty acids. They are also hydrophobic, and they play a role in the mechanical stability of the cell membrane. We've done this last time, लेकिन फिर से हम अभी कर रहे हैं. क्या मतलब mechanical stability? मतलब if the temperature is too high, cholesterol will prevent the cell membrane from melting, from becoming more fluid. If the temperature is too low, cholesterol will prevent the cell membrane from becoming too solid. So it's going to play a role in the mechanical stability. एक और चीज क्या? कि the cholesterol, since it's a hydrophobic substance, so it acts as a barrier to the movement of many polar substances. तो इसका मतलब ये हुआ कि बहुत सारे ऐसे polar substances which might be toxic to the cell और जिनको cell के अंदर नहीं आना चाहिए, तो cholesterol और जो ये hydrophobic fatty acids हैं वो उसको रोकेंगे. Similarly, there are many water-soluble substances जो cell के अंदर हैं. They should be remaining inside the cell, so they will not be allowed to leave the cell. So essentially, the fact that the cell membrane is a hydrophobic structure plays a very important role in retaining some important water-soluble substances inside and also not allowing some toxic water-soluble substances to come inside. Number one. Number two, what about those substances which are water-soluble, polar, and very useful, and they still can't enter? So for those, we have these proteins, which you can see over here. Okay? ये भी एक protein है. This is also a protein. So we have different kinds of these proteins which are found on the cell membrane. Either these proteins can completely span the membrane. Span का मतलब क्या? What do you mean by span? जी आमना बताओ कि if I'm saying that this protein is spanning the phospholipid bilayer, what do you mean by that?
Just making it appear bigger.
Not bigger. Span means कि completely covering the phospholipid bilayer. ठीक है? Cover कर रहा है पूरा का पूरा. For example, देखो आपको यहाँ पे कितनी proteins नजर आ रही है ना? There is this protein which is completely covering the phospholipid bilayer. This protein is not covering. It's only found on the inner part of the phospholipid bilayer. This protein is found on the outer part. ये पूरा span कर रही है. ठीक है? अब समझ आ रही है बात?
Yes, miss.
We have different kinds of these proteins. So either they can be just found on the exterior, ठीक है? Or they can be just found on the interior, or they can cover the entire phospholipid bilayer. And different kinds of proteins have different functions. Let's take them one by one. Here you can see this is a transport protein. मतलब कि ये एक ऐसा protein है कि जो since this is a hydrophobic core और ये नहीं allow कर रहा water soluble substances को pass करने के लिए, तो let's say अगर glucose को अंदर आना है, glucose wants to come inside, so it will come through this transport protein because it's, let me rub this and tell you कैसे glucose आ सकता है, because जो इसका ये वाला part है ना, I'm just coloring this part. This part is hydrophilic and this part is hydrophobic. ठीक है? क्या मतलब हुआ इस बात का? ये तो प्रोटीन है ना, हमने प्रोटीन्स किए हैं बायोलॉजिकल मॉलिक्यूल्स में, so we studied that many proteins have a tertiary structure और वो उसमें हाइड्रोफिलिक अमाइनो एसिड्स भी हो सकते हैं, हाइड्रोफोबिक भी हो सकते हैं. तो ये भी उसी तरह का एक प्रोटीन है कि इट इज मेड अप ऑफ हाइड्रोफिलिक आर ग्रुप्स एंड हाइड्रोफोबिक आर ग्रुप्स. जो इसके हाइड्रोफिलिक आर ग्रुप्स हैं वो बिल्कुल बीच में, तो वो आराम से ग्लूकोज को अंदर आने दे देंगे, राइट? Whereas the hydrophobic part is, this part is facing the hydrophobic fatty acids इस तरफ से and this part is facing it इस तरफ से, तो आराम से ग्लूकोज अंदर आ जाएगा. ठीक? अच्छा, let's take another protein into consideration. If you see this protein very carefully, तो आपको नजर आएगा कि it's a protein that spans the phospholipid bilayer, लेकिन इस पे एक polysaccharide लगा हुआ है. Polysaccharide ही है क्योंकि ग्लूकोज 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 ग्लूकोज, इट्स अ पॉलीसैकेराइड. सो इट्स अ ग्लाइको, ये इसका ग्लाइको पार्ट है, प्रोटीन, ग्लाइको प्रोटीन. अच्छा, ग्लाइको प्रोटीन हो गया. फिर एक और चीज हमें नजर आ रही है. आई एम नाउ जूमिंग इनटू दिस. ये तो सारे आपके फॉस्फेट हेड्स हैं, है ना, और इस पे भी ये लगा हुआ है ग्लाइको यूनिट. तो व्हाट विल दिस बिकम? ग्लाइको लिपिड. लिपिड क्यों? क्योंकि ये किस पे लगा हुआ है? फॉस्फोलिपिड पे लगा हुआ है. So to summarize, I will say that these different structures which I'm talking about right now, whether it's cholesterol or glycoprotein or glycolipid or transport proteins, they have separate specific functions. They give the cell membrane a mosaic appearance. The cholesterol maintains the stability of the cell membrane. The transport protein provides a channel for the water-soluble substances to pass through. What do glycoproteins and glycolipids do? They function as cell markers or cell antigens. या फिर हम कह सकते हैं कि सेल रिकग्निशन में इनका रोल है. कैसे? ये बिल्कुल ऐसे है जैसे कि दे फंक्शन एज एन एंटेना. ये एंटेना लगा हुआ है सेल के ऊपर. थ्रू दीज एंटेनास, ये भी एक एंटेना है, ये भी एंटेना है. this cell will recognize another cell. Now, both of you, I'm giving you 2 minutes to think about how will these polysaccharide chains play a role in the recognition of one cell with the other. Or in other words, let me rephrase my question कि क्या जरूरत पड़ती है बॉडी में किसी एक सेल को इस चीज की कि वो दूसरे सेल को recognize करे? Can you think about a scenario? Yes. For example, you are, for example, this is the blood of a person and he's A positive. This is the blood of a person and he's B positive. So this means that this person has specific glycoproteins on the surface of the red blood cell, which makes him A positive. ठीक है? इसी तरह से इसके पास अपने glycoproteins हैं, which make it B positive. And if this person needs blood, and if I transfuse this blood into him, it is going to cause a reaction, an antigen-antibody reaction, and it can cause death. Okay? So then this means that these glycoproteins and glycolipids, they are also helpful in cell recognition. They behave as cell antigens, they behave as cell markers. और उसके अलावा और भी बहुत सारे इसके फंक्शन हैं. Yes. Go through this. Glycolipids and glycoproteins. Insulin is released in your body when your blood glucose is high, released by pancreas, carried by means of blood. और ये इंसुलिन को कैसे पता चला कि उसको जाके लिवर की सेल मेम्ब्रेन से अटैच होना है? So now, since you have the knowledge of the cell membrane, if I ask you in one word, which molecule out of all these molecules are playing a role for the recognition of insulin to the cell surface membrane of the liver? So what will you say? What is that molecule?
Glycoprotein.
Glycoprotein or glycolipid. It can be glycolipid also, it can be glycoprotein also. So this means that every cell is unique. Liver cell has its own receptors. और जब हम कहते हैं ना उसके पास अपना receptor है, इसका मतलब उसके पास अपना glycoprotein या glycolipid है. Why is it कि when dopamine is released in your blood, तो वो उसको पता है कि किस organ पे जाके act करना है, ठीक है? Why is it that when adrenaline is released from adrenal glands, it will dilate your pupils? ये pupil को जाके dilate करेगी, heart rate को जाके increase करेगी. इसका मतलब adrenaline की तो इतने सारे receptors हैं, right? So every chemical in your body needs where to act, and the reason why it knows where to act, and the reason why it knows where to act is through the presence of these receptors, and they are playing a very, very crucial role in your the life of the cells, right? So, if I have to summarize कि जितने भी सेल मेम्ब्रेन पे प्रोटीन लगे हैं, अब हम प्रोटीन्स की बात कर रहे हैं, ठीक है? अभी तो हमने ग्लाइकोप्रोटीन्स की भी बात की एंड ट्रांसपोर्ट प्रोटीन्स की भी बात की. बट इफ आई कंबाइन ऑल काइंड्स ऑफ प्रोटीन्स जो कि सेल मेम्ब्रेन पे इधर उधर लगी हुई हैं, एंड इफ आई हैव टू समराइज कि इनके क्या क्या फंक्शन हो सकते हैं, तो आई वुड से सबसे इंपॉर्टेंट फंक्शन है कि दे फंक्शन एज ट्रांसपोर्ट प्रोटीन्स. ठीक है? ये चैनल एंड कैरियर जो है, ये 2 काइंड्स ऑफ ट्रांसपोर्ट प्रोटीन्स हैं जो हम बाद में करेंगे. वी आर नॉट डूइंग देम टुडे. Proteins themselves can behave as enzymes. Some of the proteins, they can behave as receptors, जो हमने अभी बात की ग्लाइकोप्रोटीन की. ठीक है? इस पे ग्लाइकोप्रोटीन में ऐड कर देती हूँ. ग्लाइकोप्रोटीन, they can help in cell to cell recognition, जो हमने बात की कि phagocyte recognizing a bacterium. ठीक है? इसमें हम ये ऐड कर देते हैं. Intercellular joining, 2 cells want to attach to each other. This may be. And if you remember, if अगर आपको याद हो cell structure में, तो ये जो आपके microtubules है ना, attachment to cytoskeleton, it is also possible through these membrane proteins. कैसे? Just giving you an example. Let's say this is your cell, ठीक है? ये आपका nucleus हो गया. This is your RER, ठीक? और आपके RER से vesicles bud हो रहे हैं, and the vesicles want to go and attach to the Golgi. तो आपके पूरे जो सेल में ये माइक्रोट्यूब्यूल्स हैं, राइट, दे आर द साइटोस्केलेटन ऑफ द सेल. यहाँ भी हैं, यहाँ भी हैं, एंड दीज माइक्रोट्यूब्यूल्स दे मेक इट इजियर फॉर द वेसिकल्स टू स्लाइड एंड रीच द गोल्जी. तो अब ये कैसे जा रहे हैं? इट्स ओनली पॉसिबल थ्रू द फैक्ट कि सेल मेम्ब्रेन के पास प्रोटीन है, क्योंकि ये भी एक सेल मेम्ब्रेन है, ये भी एक सेल मेम्ब्रेन है, ठीक है? Vesicles की भी कोई cell membrane होगी, तो it's only because of the cell membranes that structures can attach to cytoskeleton, and they can also attach to some extracellular components. Guys, I hope I'm making whatever I'm explaining you is making sense. G, any questions?
No, miss.
No? Achha. Look, different kinds of membrane proteins acting as a receptor, acting as an enzyme, acting as a transport protein, a carrier protein, or a channel protein, and acting as receptors, acting as different things, right? This is basically a summary diagram of all different functions of membrane proteins. Now, we still have time, so we can do the topic of cell signaling, and I will teach you this topic from your textbook. Okay? Just now, we were discussing about why is it that the insulin can only be recognized by a liver cell. So this means that everywhere inside our body, we have different kinds of signals, okay, which are recognized by cells through the presence of cell surface receptors. These signals can be of two types, a chemical signal and an electrical signal. बहुत simple करके मैं आपको समझा रही हूँ. An electrical signal और a chemical signal. ठीक है? जैसे अगर हम इसमें ये वाली एग्जाम्पल फिट कर लेते हैं, कौन सी वाली? The example of insulin. और let's glucagon की कर लेते हैं, ठीक है? Now the stimulus is that your blood glucose is falling. Listen to this very carefully. Because you are fasting or starving. This is a stimulus, okay? You have pancreas, leaf-shaped organ. It gets stimulated and releases the hormone glucagon. As per the definition of a hormone, since hormones are chemical substances which are made by endocrine glands, तो ये आपका endocrine gland है, carried to the target organs through blood. यही आपको O level में पढ़ाया गया, right? तो अब ये blood के through कहाँ जा रहा है? Let me make blood for you. ठीक है जी, ये blood के through जा रहा है. This is a blood vessel. Through blood. To the liver. And the liver cell has the receptors for, receptors का मतलब glucagon, glycoprotein. Glycoprotein receptor. Specific for glucagon. Okay, so carried by means of the blood. So glucagon will be your what? Chemical signal. Electrical भी हो सकता है, हम अभी electrical नहीं कर रहे. We will do electrical signals in A2. ये chemical signal जाता है through the blood and goes and attaches to the receptor. Let me make glucagon. Since I'm using green color, so this is glucagon. ये जाके यहाँ पे लग गया. This is just one tiny example. Imagine multiple actions are carried out in your body. कभी प्रोटीन बन रहा है, कभी प्रोटीन टूट रहा है, कभी क्या हो रहा है, कभी क्या हो रहा है. So this means that hundreds and thousands of such signaling mechanisms are going to happen inside your body, which shows that tissues and organs and systems have an excellent way of communicating with each other. आपस में एक दूसरे से बड़ी जबरदस्त communication कर रहे हैं through these signals. Every signal has a starting point, and that starting point is a change in your body, and that change is the stimulus. The change is followed by these processes, which leads to the development of a signal, and the signal will attach to its specific glycoprotein receptor. ठीक है? अच्छा, इस signal को जो है आपकी AS के language में, the terminology that the AS syllabus demands from you, you are supposed to call this signal as a ligand. Ligand. क्यों ligand? Ligand, I think it's a Greek or a Latin word. जिसका literal meaning है to bind. So why are we calling it ligand? Because it's binding to this receptor. ठीक है? Let's go to the textbook. इतनी खामोशी है guys, girls. I hope कि आप लोगों को मेरी आवाज आ रही है.
Yes, miss.
आ रही है ना? Okay. Let me show you the cell signaling mechanism. ये रहा. आधा तो मैंने आपको समझा दिया. Look. Signal or ligand. Let's imagine or let's assume that this ligand is glucagon. Glucagon is attaching to its specific protein receptor. अभी हमने कहा कि proteins receptor के तौर पे भी act कर सकते हैं, enzyme के तौर पे भी act कर सकते हैं, ठीक है? The ligand is binding to the receptor, which brings about a change in another protein. याद है मैंने कहा था कुछ proteins span करती हैं और कुछ protein सिर्फ और सिर्फ inner part पे लगी हुई होती हैं. So this protein, which is a G protein, is attached only to the inner part of the cell surface membrane. वो part जो कि cytoplasm को face करता है, ठीक है? And the binding of the ligand to the protein receptor is going to activate the G protein. Activation of the G protein is going to activate an enzyme. अब ये enzyme भी एक protein ही है क्योंकि अभी पीछे हमने कहा कि cell membrane proteins can behave as enzymes also. So this protein activation activates an enzyme और ये जो enzyme है ना वो it makes a second messenger and the name of that second messenger is cyclic AMP and the name of the enzyme is adenyl cyclase. Alia, can you repeat what I said right now? यहाँ पे पूरा आप मुझे बताओ मैंने क्या बोला?
The protein receptor is binding to the G protein and it's activating it when it combines with the ligand.
नहीं, सबसे first क्या हुआ? What's the first thing that's
and get the cyclic?
Okay. Again, I'll repeat.
Okay, is binding with the ligand.
चलो ठीक है. आमना आप बताओ. First step.
The ligand binds to the protein receptor.
Okay. And then?
And then it signals the G protein, which activates the second messenger.
So step 1 is binding of the ligand to the protein receptor. Step 2 is activation of G protein. Step 3 is?
The second messenger is made because of the activation.
Activation of what?
It's activation of an enzyme.
What's the name of the enzyme?
Adenylylase.
Cyclase, sorry. Sorry for the spelling mistake. Adenyl cyclase. Adenyl cyclase is the enzyme, and the second messenger is cyclic AMP. Okay, you can mute yourself. Since 5 minutes are left, so listen to this part very carefully. The second messenger is activated, ठीक है? और फिर क्या करता है कि ये एक सेकंड मैसेंजर का मॉलिक्यूल नहीं है. वी हैव many such molecules जो यहाँ पे आपको नजर आ रहे हैं, छोटे छोटे पर्पल कलर के. These ones, we have lots of these. All these second messenger molecules will activate not one enzyme, 1, 2, 3, 4, 5, 6, हो सकता है इससे भी ज्यादा हो. And these enzymes will activate other enzymes. और वो सारे enzymes जो है ना, वो रिजल्ट देंगे जो आपको चाहिए इस वक्त. फॉर एग्जाम्पल, अगर ये ग्लूकागॉन है, तो वी नीड द ग्लूकागॉन टू कॉज अ मेटाबॉलिक चेंज, एंड दैट मेटाबॉलिक चेंज है कि सिंस आपने रोजा रखा हुआ है या आप फास्टिंग कर रहे हो, तो आपको क्या मेटाबॉलिक चेंज चाहिए? आपको लिवर के अंदर जो ग्लाइकोजन है उसको ग्लूकोज में तोड़ना है ताकि योर ब्लड ग्लूकोज रिमेन्स स्टेबल. तो योर डिजायर्ड एक्शन इज मेटाबॉलिक चेंज. तो मेटाबॉलिक चेंज के लिए जिस एंजाइम की एक्टिवेशन की जरूरत है, those will be activated. And these are the other examples given to you. So, to sum up, I can say cell signaling is a mechanism by means of which different cells, tissues, and organs, they receive signals from each other so as to carry out a desired action. The initial signal is just one molecule, but it results in the amplification of the original signal. How? Because the initial signal was one ligand, it led to the formation of many second messenger molecules. क्या नाम है second messenger का? Cyclic AMP. Those many second messenger molecules activated many enzymes, और उन many enzymes ने और ज़्यादा enzymes को activate किया. तो क्या हुआ? The cascade that had started with one ligand is amplified at each stage, and hence this cascade pathway is known as a signaling amplification cascade pathway. इसको हम कहते हैं amplification cascade. कैसे किस्म के questions आएंगे exams में? You will be given an unknown situation. कुछ भी, कोई भी example, अभी मैंने ग्लूकागॉन की दी है, कुछ भी दे सकते हैं आपको वो, and they will ask you to talk about cell signaling mechanism in the unknown situation. अच्छा, अब हम क्या करते हैं कि I would want you to take a screenshot of this. हाँ, ठीक है? I'm just waiting. Please take a screenshot of this. इसके 3 marks हैं, ठीक है? Take a screenshot and do this for Wednesday, both of you. This will be a practice for the concept of cell signaling.